Hello everyone, to another week on the seminars of the EBD. Today we have Dr. Tano Gutiérrez. Thank you very much for accepting our invitation to talk here.
Thank you.
And I'm going to talk a little bit about Tano. He obtained his PhD in ecology by the University of Murcia in 2014. And then as a postdoc, he has worked at Cardiff University at the UK, at the University of Barcelona, at the University of Minho in Portugal, and now here at the EBD, where he works as a Juan de la Cierva Incorporación. It's a very nice, very well-rounded career. As an ecologist, Tano investigates global change impacts on both biodiversity and ecosystem functioning, and in both freshwater and terrestrial ecosystems. He seeks to provide scientific basis to reverse ecosystem degradation and ensure their sustainability. And for that, he combines field observations with experiments and meta-analysis. So, thank you very much. I'm going to pass you the word. Tano, it's all yours.
Thank you very much for the presentation. And thank you very much for coming here and also for the people online. For me, it's a pleasure to have some time to show you some of my research and to hear your feedback. And today we are going to speak a little bit about some type of ecosystems, rivers, that probably we don't work that much with these ecosystems in the station, but I think it's a great opportunity also to view these ecosystems from your angle, and the idea is to see this new ecosystem. So typically we care about rivers when we have a disaster, when we have a really bad situation, and normally it's too late, and management options to restore ecosystems and to fix the problems are limited and expensive. This is one very paradigmatic example. This is the Marmala Lagoon in Murcia, in the southeast of Spain, and here you see like a massive flood. And if you compare the color of the Marmala Lagoon with the Mediterranean Sea, you can imagine what's going on, okay? But this is probably a long story and not caused by one single stressor or one single human impact. And this is what I want to show you today. A lot of scientific publications were warning about, we are accumulating stressors, we are having a lot of agriculture. You can see here is basically all the catchment is full of intensive agriculture. More or less, probably it's a similar situation, what is happening in Doñana, with the difference that here they are not taking water because the soil is so saline, but the intensification is pretty similar. And the thing is, during 20 years, from different institutions, they've been worrying about, we are having too much agriculture, but managers, they say, but water is still etc., but they were not considering, for example, in this case, climate change or other pressures that in the end combined to do a very sad ending. So now to solve this problem, probably we need to spend a lot of money in the best case scenario, and we don't have anything clear if we are going to get or not a good result. So the thing with the fluvial ecosystems, rivers, streams, and also other type of freshwater ecosystems like lakes, where I work less often, the thing with these ecosystems is that they are subject to a range of pressures which normally correspond to the basin, to the catchment where they receive water. For example, here, imagine if we are, for example, in the mouth of the river, we can be very far from some places where the impact is taking place. Imagine we have here agriculture or here agriculture. This agriculture, despite being hundreds of kilometers far from the mouth, they can be having an impact. For example, in the Guadalquivir River, here in Seville, most of the impacts, most of the problems come from headwaters. So this is a situation which is a little bit different from other terrestrial ecosystems and require a different management. And in terms of the rivers, we need to take care about what's happening in the catchment. And for this reason, rivers are sensitive to a wide variety of stressors, being, for example, regional stressors or even global stressors. And another interesting aspect of rivers compared to terrestrial ecosystems that they normally have a negative net productivity. And why this is happening? Well, if we look at, for example, forests, grasslands, or agroecosystems, normally they have positive net production. They produce basically a lot of plant organic matter, and this is the basis for the food webs. But what happens if we come back to the other slide is that all this production naturally ends on rivers and freshwater ecosystem, because rivers normally are in depressions, are in the drainage system. So this make rivers very important in terms of transforming, processing, and consuming organic matter. And for this reason, roughly half of the terrestrial production that reach rivers is transformed to CO2 or other type of greenhouse gases in a way that only half of this amount of organic matter reaches the ocean. So this is important in several ways. First, this is very important in terms of the global carbon cycles and climate change, and also it's very important for marine productivity. So rivers in this sense are important for other ecosystems, and of course preserving their health will be important beyond their own status. What happens now, as I was telling you, is that freshwaters are generally in a very bad condition across Europe. If we look at the general picture, roughly 60% of the European surface waters, including rivers, are not in, they don't achieve the legal good ecological status, which means that they fail to achieve some, for example, to have some type of animals, or they have some problems, for example, with toxic substances. And to be honest, I don't trust very much the map here. I would say, I would put more red here in the south. But anyway, this is what we get. Anyway, the picture is not looking very good. But the thing is, we don't know very well, first, how to restore these ecosystems, and second, the consequences of river degradation. So one of the ways to address this problem is understanding which stressor combinations, which combination of pressures are triggering river degradation. And I was working in the European Mars project some time ago when I was working at Cardiff University in the UK. And one of the key figures of this project was understanding that most of the river ecosystems were affected by more than one pressure. This means that most of these red colors probably are the result of having, for example, a lot of nutrients in water, of having flow regulation, invasive species, combination of things. So managers, they struggle to understand which mitigation option they should take to transform this red into blue. This is the key question, and which is the most, let's say, cost-effective action. And this was one of the key goals of the Mars project. So the combination of these global chain pressures in the real world of the river, they act locally, and they transform in they modify different local conditions. For example, if we think about agriculture or land use intensification in general, they normally result in an increase of nutrient concentrations, also in a decrease of oxygen, dissolved oxygen, which is when compared, for example, with terrestrial ecosystems, is one of the main limitations, because in terrestrial ecosystem normally you don't have problems of anoxia, but in waters this is very important, and you can have, for example, if you have a lot of nutrients, in the end probably you are going to have anoxic conditions, and This is what happened, for example, in the Marmala Lagoon. Also, we have warmer waters, we have more pathogens, and you have alterations in flowing conditions, which is, let's say, the key element of rivers, of running waters. And the combination of all these stressors, they are causing an unprecedented change in river biodiversity. And the thing is, as I was telling you, we don't know very well the consequences of this change. One of the, let's say, the big themes of this biodiversity change are mainly vertebrates, and we have really dramatic losses of freshwater vertebrates, for example, especially for fish species. Most species have declined by 80, the average is 83% since the 70s, which is a lot. And this is crucial evidence showing that rivers are suffering global change more intensively than, for example, terrestrial or marine ecosystems. The key question now is understanding all these contributions to global biogeochemical cycles. Most of them are mediated by complex food web networks. And the thing is understanding if we are modifying biodiversity in the different parts of the river, how this will affect, for example, to greenhouse gas emissions, to marine productivity, and the other functions provided by rivers. So the studies I will show you in the next slides will try to understand small portions, very small portions of this picture. So basically, the goal of my studies is understanding how global change, in the end, translates to biodiversity and ecosystem function changes. So rivers are home of a disproportionate biodiversity compared to their surface, and this is good in terms of, okay, we have a lot of elements to evaluate biodiversity change. So I will show you some of them. I will work with some of these elements today. I'm not a specialist in any of them, but a little bit more in macroinvertebrates. But I think the strength of some of these approaches are trying to combine these different trophic levels and to understand what happens from a multi-trophic perspective. The core idea of most of these studies is, okay, as long as we have more biodiversity and more trophic levels, we will get more stable and higher amounts of ecosystem functioning. And this occurs because I will give a brief overview of how river food webs work to understand more or less this biodiversity ecosystem functioning relationship. So rivers, normally they have two types of primary producers. They have aquatic primary producers, basically algae and microphytes, both microalgae or macroalgae. And you have surrounding rivers, you have plenty normally of woody vegetation, riparian trees. So these are the two basal sources of the food web. Then we have microbial decomposers, which play a key role. As I was telling you, rivers are mainly heterotrophic. So decomposers play a key role in terms of recycling nutrients and organic matter. And then we have the intermediate trophic level, which are invertebrates, with basically they can feed on a wide range of resources, either directly from aquatic producers or indirectly leaves mediated by decomposers. Decomposers here play a key role because leaves, the problem with leaves happens the same in, for example, in terrestrial ecosystems. Leaves are very difficult to process because they have a lot of carbon relative to the nutrients, normally to nitrogen or phosphorus. So fungi and bacteria, mainly fungi, but also bacteria, they concentrate the amount of nutrients, making them more suitable for some type of invertebrates. But the thing here is invertebrates can concentrate energy and matter, And basically, they are the source for top predators, which normally are the animals we know from rivers: birds, fishes, amphibians, and others. So the idea is understanding the different characteristics of these food webs to predict how changes in biodiversity will affect to ecosystem functioning. So in this first paper, our question was understanding which invertebrate characteristics. As I show you here, we have different characteristics. We have the richness, the abundance, on also looking at different functional traits of the invertebrates. So our question here was understanding how the different configurations of the prey, how these result in different amounts of culturally valued birds or fishes. Basically, we were working with with the dipper, grey wagtail, and Atlantic salmon and trout, and brown trout in the UK. So our hypothesis here, we tested 4 hypotheses. I will show you 4, I think it's more interesting. The first one was that some key players, some key prey will be sustaining the 4 predators because they, for example, they are easy to catch. For example, they are more vulnerable. A second alternative hypothesis was that the diversity of prey, the functional diversity of prey having, for example, different sizes, different ways of reproduce, occupying different habitats, this will be beneficial because, for example, this will favor niche partitioning for predators and will increase their productivity. Another hypothesis was the functional equivalent of the prey, which is they don't care about what they are eating, they just care about the amount. And finally, some kind of null hypothesis was that abiotic factors like, for example, pH or elevation are driving the whole thing irrespectively of biodiversity. What we do here is basically the first thing we did was trying to understand how prey differ in terms of their functional traits. So we classify 4 types of prey, basically in relation with their habitat. For example, prey that occur in fast flow and they are present in the drift, and others that typically they prefer, for example, to live in the substrate, or they can swim, and also regarding their body size. And what we found here, in terms of these 4 hypotheses, is that some animals, some prey, having specific traits were the most important to sustain the four populations. We identified that the abundance of a key functional group, in this case small organisms that normally they are present in the drift, so they don't know how to swim, they just go with the flow. This type of organisms are critical to sustain the 4 predators. So in this way we can understand if we lose, for example, this type of prey, we will get an immediate loss of vertebrate predators. So this is somehow a way to understand, for example, why some iconic predators are disappearing. Here we change absolutely the focal organisms, and we focus on aquatic fungi to understand how one stressor, in this case when the rivers become dry because of climate change or even naturally, we wanted to understand how this pressure will translate to some functions delivered by fungi, which is basically decomposing organic matter cycling, and also producing fungal biomass. Fungal biomass is important because it's the food for the invertebrates, for some type of invertebrates. So we combined a field and a manipulative experiment to explore how these stressors can alter the way in which biodiversity, fungal biodiversity, influences these 2 processes. So what we found here is that in both cases, having more fungal richness have a positive effect in both functions, in organic matter decomposition and in fungal biomass accumulation, and that the effect of flow intermittence of rivers becoming dry was mediated by fungal biodiversity, which means that the real driver of the change is the biodiversity change. Losing fungal biodiversity will translate into reducing the composition rates and fungal biomass accumulation. Okay? And we also perform an experimental design to confirm a little bit what we found in the field, and also to test if biodiversity have a buffer capacity to counteract drying conditions. So in this case, we apply some drying treatments. This is like the control. We have a flowing river. We have a river with a short non-flowing period, and a river with a long non-flowing period. And also we have treatments with high fungal richness and with low fungal richness. What we found here is that the high richness treatment always show a higher level of organic matter decomposition, and the same for fungal biomass, which means that rivers with more biodiversity of fungi will be able to cope a little bit better with climate change. And here we look, we did a similar study but with invertebrates. Our key question here was, okay, we have some evidence that drying stress, when rivers become dry, normally these rivers have a lower amount of animals. They have less invertebrate biodiversity and lower abundance. But we don't know very well the mechanisms linking these two things, the diversity and the abundance. So our hypothesis here was that drying may have a direct effect on both. Basically, for example, through abiotic filtering, some organisms cannot complete for example, their life cycle, and also they can have direct effects in terms of performance. They can live there, but they can grow less efficiently. But we also wanted to test a diversity effect which will link diversity and density. This is that, for example, rivers having more biodiversity of invertebrates will show more possibilities of having niche complementarity or facilitation interactions in a way that having more biodiversity will benefit invertebrate density. So our idea was testing these direct and indirect mechanisms to show how drying stress can reduce density. And what we found here, testing different structural equation modeling, what we found is that basically similar to the other fungal study, we found that the effects of drying stress characterized through different metrics, the annual duration or the frequency, basically the effect is mediated again by either taxonomic or functional diversity of invertebrates, which means that, again, the loss of invertebrate production or invertebrate density is mediated by their biodiversity. Rivers having less biodiversity of invertebrates will tend to have less density, and climate change can increase this effect. So now I want to give a brief overview of the multiple stressor world. So until here, we've been exploring what happens if one stressor alters biodiversity and exploring the functional consequences. But for example, as we saw at the beginning of the presentation with the Marmala example, in the real world ecosystems, we don't have one stressor, we have multiple stressors. We can have climate change, we have warming from climate change, we can have nitrates, etc. And here we have it's a little bit more complex to understand biological and ecosystem responses. So this is just an example, and it's a little bit it's not the best, probably the easiest thing to understand, but when stressors combine, can give rise to different type of combined effects. For example, in this case, we have probably the simplest case, which is an additive effect. So imagine, for example, the yellow line could be like the baseline, and we have for example, sorry, I will start again. We have two stressors, stressor 1 and stressor 2, and depending on the levels of both stressors, we will have more or less degradation respect, for example, to an indicator. So, for example, the red line illustrates the maximum degree of degradation, and in this case, we have a higher value of the second stressor and a higher value of the first stressor. And the same applies to the other lines. Okay? So, the less level we have in the second stressor, the less degradation we have. Basically, this is the sum of both effects. Okay? Probably the way in which I explained this wasn't the best, but basically here we have the sum of both stressor effects. And on the other side, we have a completely different story. We have a non-additive effect. In some cases, the final effect of both stressors could be less than the sum of both stressors, which is the antagonistic effect, or the other way around, we can have both stressors amplify their effects, and in the end we have a much higher effect. Okay? I don't want to go much into details, but basically to understand that when we have more than one stressor, ecological responses can be more complicated, and we need to consider that. And understanding which effect we have is key for mitigation, and I will show you why. For example, imagine we have nitrates in one river and warming. Imagine this is Marmala Lagoon. Okay? We can have different combined effects. The first one is, okay, only nitrates matter. Okay? In this case, we should care about only one stressor. Imagine we have an additive effect. Okay? We should care about both, but basically they can operate in highly predictable conditions. We can have a synergistic effect. Okay, in this case, when we have high values of both, this is problematic. And we can have an antagonistic, which means both counteract at some point, but also we need to take care about that. So if we are a manager, we need to know how these stressors combine to adopt different mitigation actions. For example, in these cases, let's say not always the optimum, but a good strategy could be tackle the dominant one. Okay, because in the three cases, if we reduce the level of the dominant one, imagine nitrates, if we reduce nitrates in three cases, we will get an ecological benefit. But here, for example, with antagonistic situations, if we reduce nitrate, this 1.5 can increase. It seems a little bit, let's say, not very intuitive, but it can happen. And this is what's happening in many cases. So this is the interest of understanding the combined effect of multiple stressors. So I will show you some results. I don't know how it's going with the time. Okay. I will show you some meta-analysis and review works where we try to synthesize which type of combined effects are more common in freshwaters, which is, it's essential to understand which mitigation actions should be preferred. So here I will show you some meta-analysis based on purely manipulative experiments and other combining both. Why I'm doing this distinction? Okay, because you will see that things can change. Manipulative experiments, as I showed you, for example, with the fungi, are very useful to understand the causal effects of stressors. We can mix, for example, nitrates and warming and understand what's happening, for example, for algae or for other type of organisms. Okay, but normally conditions are not always realistic. And when we go to the field and do the same experiment, we have more realistic conditions, but sometimes we don't know really if our two stressors are the main cause or if we have another confounding effect. So the idea of this combined meta-analysis was to understand the pros and cons of both methods. So what we see here is that interactive effects, as I was telling you, synergistic or antagonistic effects, for example, are common in this meta-analysis, which was performed in marine ecosystems, but in freshwater ecosystems, which are these three meta-analyses, normally we have a mixture between non-interactive and interactive responses. So for managers, this is a little bit good news because they can have, they will have less often complicated situations, but we still have some cases where we have interactions. So when we look to the percentage of the interactions, we see that in freshwater ecosystems, antagonistic interactions are more common than synergistic interactions. And this is curious because when you look at most of the multi-stressor literature, one of the warning messages was, okay, if we have two stressors, they can have synergistic effects. But in reality, this is happening less often. Okay? This is good in terms, okay, when we have more stressors, the final negative effect is less than expected. Okay, this is good, and there are some ecological reasons for that. But in terms of management, this is highly complicated to restore because reducing one stressor level can even increase the final effect. And this is the bad news. We have some good news and some bad news, okay, regarding this. And another interesting thing is that this situation can change depending on the ecosystem. For example, in a pan-European study we saw that, for example, in lakes, interactions between stressors are rare, but in rivers can be very important in some cases. So basically, the conclusions of the current knowledge is that we have a poor understanding of how multiple stressors influence ecosystem function. We know how single stressors can affect, and especially at large scales. At large scales, we know very little. We have manipulative studies or regional studies. Also, we know very little how the combination of multiple trophic levels and the characteristics of trophic webs, how this translates into ecosystem functioning changes and how these properties respond to stress, we still know very little. It seems that combining both manipulative and observational studies could be a good solution for that, but as I say, now most of the literature is based on manipulative studies. So basically the gap is here. And also to understand, for example, when we saw the initial slides, when we saw the catchment, the river, and the sea, now we have these small pieces of the story, but there's a need to scale up all these things to understand how, for example, changes in local biodiversity will translate to different biochemical cycles and, for example, greenhouse gas emissions and all these things. So these challenges are the core idea of a new project I started recently. Well, I started officially recently, but I was working in this at least 1 year. And the idea is building or creating an Iberian observatory to monitor large-scale and long-term changes in river biodiversity and ecosystem functioning. So the idea is understanding, for example, if, for example, current legal limits of nutrients should be changed or should be reduced. For example, if we have warmer conditions or if we have invasive species. For example, now current legal limits are about 25 milligrams per liter for nitrates, okay? But will be this efficient to, for example, to stop or to prevent eutrophication if waters become warmer or we have more invasive species? So this is a question that needs to be addressed. And to be addressed, I think, we have some information from manipulative experiments, but we need to look at real ecosystems to see these things. And also we need to integrate different trophic levels from microbes to vertebrates and different ecosystem functions. So the idea is trying to integrate this whole thing to respond to these pressing questions. And to do that, I did the crazy thing of gathering 70 researchers across the different parts of the Iberian Peninsula. I don't know if it's more crazy than efficient, but I started to do that, and the other day, this week, we gathered here in the same place as you are sitting now, and we started to work. This year, we are planning to start some field works, and the idea is, over the time, during the next year, consolidating this observatory and trying to understand how these things change over the long term. Now we are working with 6 areas across the Iberian Peninsula, covering an aridity gradient. We have some arid catchments from the south. The most arid one is the Segura River Basin between Murcia and Albacete. Then we have some rivers from Guadalquivir, Tinto and Odiel, basically Seville, Huelva, and Cordoba provinces. Then we have also some cold Mediterranean climate in Avila, Segovia. This is real Mediterranean climate, but the winter is much colder than in the other places. Then we have wet Mediterranean here in the south of Gredos Mountains. We have also Atlantic climate with a marked dry season in North Portugal, and more, let's say, regular temperate climate, humid climate in Picos de Europa. So the idea is we have different researchers from different parts of the Iberian Peninsula. Some of them were going to the field to collect samples, and others will process the different samples. So the idea of this observatory is, as I said, trying to understand long-term changes in rivers and the impacts of different global change drivers. Also, the idea is creating a network of people looking at the same thing but from different angles, and to exchange expertises, to try to produce something more integrative, and finally to provide some kind of, let's say, experimental setup to add more questions in the future. This is not really a specific goal. This is not exactly the idea, but this is some type of questions that we can address with these data sets, and currently with our current information, with our current knowledge, we are unable to give answers to this. For example, understanding how multiple stressor effects, how these arise at, how these appear at large scales, if they are consistent or not. For example, understanding if the effects of biodiversity on ecosystem functioning change over time, and if they change depending on stressor combinations. For example, understanding if rivers produce more or less greenhouse gas emissions with warmer conditions or not. For example, we are looking now at this, and we are seeing that polluted rivers are sources of greenhouse gas emissions, which is also interesting and a new thing, but we don't know how this changes over time. And another interesting thing is monitoring how microbial and insect populations change over time. The thing is, for example, for vertebrates, we have information. For freshwater fish, we have information also for other organisms, but for insects and microbes, we know very little. And they have a key role on the different functions delivered by rivers. So this is just a picture of the meeting. This week was a little bit intense. To be honest, I'm probably, this is not my best day to give a talk. To be honest, I'm quite unfocused, and probably I didn't have enough time to prepare this, but hope you enjoy it, and it was stimulating for you. Thank you very much.
Well, yeah, thanks anyways for agreeing to give a talk this week then.
Thank you.
We really, really appreciate it. And it's really fantastic to see these really big collaborative efforts. Any questions?
Thanks. Just I wasn't sure that when you say that the key praise in the river ecosystems were very important because they go with the flow of waters, and so the predators can eat them at all time, and so that's why they were so important. Is that what you said?
Yeah, this is one trait that make a lot of insects very vulnerable to predators because, for example, trouts and salmons, the juveniles, they normally eat against the current. So they open the mouth, and basically they are engulfing a lot of invertebrates. So the thing was understanding which traits make them more vulnerable to prey, to make the study more general. For example, this took place in the UK, but some of the insights can apply, for example, to the Iberian Peninsula or to other European regions. And the idea was using traits precisely to make it more general. But for example, in this case, these traits make them vulnerable to predators.
Okay. Well, thanks.
You're welcome.
Thanks, Tano, for the talk. I was wondering, also in part because of Miguel Clavero's work, we've learned over the years that our perception of what constitutes a baseline of a given ecosystem changes. So if you're planning on building this observatory to test for climate or global change, I was wondering, what kind of data do you have, and how far back can you go to compare the upcoming changes to what? Well, I mean, in other words, where are you going to consider your background baseline? What are you going to be comparing the changes with?
Well, this is a super good question because now to evaluate, for example, the ecological status, the water managers, they use what they call reference conditions. So basically, 15, 20 years ago, they did studies across Europe, and this is regarding the Water Framework Directive. If you go to the US, it's a similar story, or to Australia, it's a similar story. So they went to unpolluted rivers, and they saw, imagine, for example, okay, we have an indicator which equals 100 in the rivers. So if you are below 100, this river is polluted. But for example, this was made 20 years ago. This was the baseline, for example, for 2005. But is it valid today? I don't know. In terms of biomonitoring, this is the legal baseline, but probably it's not good because 15 years ago the climate was yet different. So probably the idea is using some kind of shifting baseline, making it probably contingent to climate or to previous conditions. So the idea is not comparing against. Then the idea is creating now a baseline. For example, this year we start. This is our baseline, and see how things evolve over time. First thing. Second thing is we have this information from the Water Framework Directive, which this allows us a little bit to understand what happened in the last 15 years. Not with the same precision we're going to see in the observatory, but it will give some information. But in terms of the baseline, probably it should be contingent to climate and other changing factors.
Well, thanks. Well, this is not my field of expertise, but I've understood what you are doing, and I like the ambition of setting up this observatory. But there's something I don't understand. When you say that something, a factor, a treatment, affects ecosystem functioning or influences ecosystem functioning, how do you quantify ecosystem functioning? So how can you know that something creates a deviation of the standard or baseline functioning?
For example, it depends if you are doing a manipulative experiment or if you are in the field working with observational data. For example, in the case where we work with fungi, we have some control treatments, and we compare, for example, the treatments exposed to drying condition against this control. This was the deviation against the control. In this case, you have a flowing condition rivers, and you compare what happens if the river becomes a little bit dry or drier. So in this case, it was against control conditions, like conventional experimental study. In the field, you try to do the same. For example, you are working over gradients normally. For example, if you are working with the observatory data, you have gradients of, for example, nutrients or land use intensification, or for example, invasive species or temperature. So basically, you are not observing a deviation. You are just understanding how, for example, your predictor relates with the function. So it can be, in some cases, for example, with nutrients, some functions, they have a ham-shaped relationship. So it depends of if you are working in an experimental setup or observational setup. In the case of the observatory, our goal was doing like predictive models as well as we can. So relating a factor or different stressors, different factors with the different functions or the aggregation of multiple functions.
It can be a semantic issue, but you know, when I see the effects of something on something, you know. But anyway, thanks.
Okay, thank you.
So there was a question by Miguel and then the other Miguel.
Okay, thanks, Cayetano. I think it's that the study of aquatic ecosystems, aquatic environments are crucial to analyze global warming and the effects on organism and biodiversity in general. No? Especially because the abiotic factors are affecting very tightly to the organisms, to the freshwater organism. But I feel that in your framework, probably there is a lack of analyzing other functions, for instance, the physiological function, especially because in a warming world, to consider which are cool lower in species clades or trophic levels versus warm lower groups are important. Well, I think is maybe important. I wanted to know your opinion or what's your idea if it's necessary to look at this physiological diversity.
No. No, this is a good one. I mean, but it depends on the scale and the organizational level. For example, we are looking things now at community or food web or ecosystem level. It's more some kind of individual level or population level. I think we currently we don't have the capacity to do that, because this is more, for example, if you focus, for example, on a species or 2 or 3 species, you can do this kind of more detailed physiological studies, or I mean, could be more than 2, 3, whatever, but a reduced number of species or trophic groups. So I think currently in the observatory we are not planning to do that, but probably in the future we can pick up a subset of organisms to do this kind of manipulative experiments. For example, in the work where in the group where I did my PhD, some people did that for aquatic beetles. Basically, they were understanding how different combinations of temperature or desiccation, how this influence their performance and their physiology. But scaling up this into community or ecosystem levels is probably a little bit difficult. So we are looking at this probably indirectly. We are seeing it, okay, if we are losing some kind of species or some type of species, probably it's because they cannot cope with the conditions because of the physiology. So the idea, for example, in the observatory is to look into this a little bit using proxies like, for example, functional traits or functional groups. You can aggregate organisms into functional groups, and some of them, they share some physiological responses or some physiological traits. And of course, it's not very detailed and not very precise, but it can give you some kind of it can tell you a little bit of the story. Thank you.
Hola, Tano. Great the presentation and great that you start the observatory. So great to do that work.
Thank you.
First, one comment about the baseline for all the work for the Water Framework Directive, where all the baselines were completely shifted because there's reference sites were only mountain streams. So basically, we don't have a baseline for lowland or middle elevation rivers. It was bad thought, I think, for the beginning, but it was difficult scenarios to plan it. So, but then I have a question for the observatory, because in there's within river there are different traditions. Then we don't make a lot of river stuff here, but also in Murcia here and in Murcia there is a lot of tradition on biodiversity, on valuing biodiversity, of the variety of life and the conservation of that variety at different scales. But then the other tradition in rivers is about bioindication, that is linked to biodiversity, but it's not the same. And it has been the mainstream in river research, or applied river research, I would say. And how is your idea of the Observatory planning to merge these two views?
Yeah, this is this is one beautiful challenge. The thing is, as you said, researchers looking at river biodiversity, they look things probably from a wider perspective. For example, looking at microbes or looking some different details. And in biomonitoring, basically, your goal is calculating an index, and you don't care about other things. And sometimes even in the index, it depends on the index consider different taxonomic levels, or even the index can ignore certain organisms, just focuses on sensitive species. So the idea of the observatory is also trying to provide a critical perspective to carry in biomonitoring. And I'm thinking in one crucial aspect, which is microbes. The thing is, for example, Water Framework Directive now is focused in surface water, is focused on diatoms, microphytes, invertebrates, fishes, and other, let's say, more structural, like the structure of the riparian vegetation and the chemical conditions of the water. But the thing is, as biogeochemistry evolves, I mean, there is increasing evidence that microbes are very important for greenhouse gas emissions, for water cleaning capacity, for a lot of stuff. So I think, and now with the metavar coding tools, it's much easier probably to get microbial richness, that invertebrate richness through conventional, or like doing electrical fishing with the fish, etc. So I think this is one of the ideas of trying to see, we have the Water Framework Directive indicators, we have more biodiversity indicators. Let's see if we can add some of the more biodiversity indicators to the conventional pool to improve the whole thing. This is one thing. And coming back a little bit to the baselines, yeah, I forget to say what you said. For some rivers, you don't have reference conditions. For example, for large rivers, you don't have reference conditions. This is the idea also of the observatory, of trying to avoid this probably comparison against the reference conditions, and probably making more dependent of key factors through continuous model. But I think this is one of the challenges, trying to convince water managers to use some novel indicators. But I know it will be difficult or impossible, but we're going to try.
So one more question over here.
Yep. Thank you. Thank you, Tano, for your talk. Well, I would like to raise a little bit of debate here. I think while I like a lot this approach, I think that it kind of contradicts a couple of the very first results that you showed us. One was that the species identity was one of the most important effects in the ecosystem functioning, and the second one was there was some sort of a hump-shaped and a saturation relationship between species richness and function too, which is, well, this is established now, right? At the light of these two results, shouldn't we try to develop approaches more directed to the species than to the whole communities and populations?
Well, again, are two different scales. Probably what happens with some of the studies I saw, I mean, some co-author studies, so it's a critique toward my studies. The thing is, when you focus on a function or small number of functions, you can be biased in terms of, for example, if you say, you're talking to a manager, and the manager tells you, I want to increase the number of birds and fishes in my river because this will attract ecotourism, blah, blah, blah. You say, you just need to increase the amount of invertebrates that drift into the river and they are small, or they are in the... Okay, if you do that, you're ignoring the consequences for other functions. So one of the main limitations of previous studies is they focus on single biological groups or single functions. So the idea is trying to understand what happens simultaneously to different functions. So if you optimize one, you may lose a lot from the others. So for example, in this paper we did some analysis to see, for example, how the different amounts of prey, if they can optimize one population of the, for example, one bird population against the other, and what happens. You see, if you, I mean, as long as you try to optimize one function, you are going to lose probably most of the others. So the idea is trying to identify how variations in biodiversity change simultaneously different functions, and from a management perspective, if you have some specific goals for some functions, for example, fish production, you need to take care about what you are doing, because you can affect to, for example, water purification capacity or thermal regulation, greenhouse gas emissions. So the idea is of the observatory is trying to address this question by integrating different functions, covering different dimensions of the multifunctionality, which is probably the most updated approach for ecosystem functioning.
What I was wondering is, if we have some tenets in functional biodiversity of the saturating relationship and the effect of dominant species, why don't we try to focus on these few dominants that are making the largest contribution to ecosystem functioning, in a way that we simplify the management of the ecosystems in order to optimize any of these functions that you are exposing here, right?
Yeah, but this will work for this specific function, but it might not work for others. This is the thing. So you cannot probably, as a manager, if you are only focused on fish production, this will work, probably. But this can work. But you are ignoring the lateral consequences for the collateral damages for other functions. If you increase a lot this, maybe these organisms are competing with others that do other functions, and you are losing other functions. The thing is, when you work, as I did, when you work with single functions, you need to be cautious in terms of your suggestions for management, because you are ignoring 90% of the story. I mean, this is necessary. I mean, this reductionism is necessary to understand how this is working. But you need to integrate different functions and to see, okay, if I modify this biodiversity aspect, how this influences the different functions. This is the thing.
That's it. I totally agree. I think that we totally need the data from the different levels of organization. Otherwise, we don't have the full picture. Yeah. So, but thank you very much, and congratulations for the observatory.
Any more questions in the room? Nothing in the chat. Lots of applause and praise.
Thank you.
So yeah, let's leave it there, right? So thanks so much again, Tano, and good luck with your monster project.
Thank you very much, Chris. Thank you.